Okay, yeah, it's absolutely beautiful to be here. This spectacular building, which I've never, in a part of Paris that I've never been to before. And this is a fantastic setting. And I take the opportunity today to tell you about how in my lab we study the assembly and the function of bacterial communities, which are known as biofilms. And the way that we approach this topic is by combining approaches from physics and mathematics with regular molecular microbiology techniques. So why do we care about these bacterial communities, these biofilms? A couple of, 4 years ago now, Hans-Kurt Fleming and Stefan Wurz, they took a census of microbial life on Earth. And microbial communities and microbes in general are found in many habitats. For example, inside us, in the intestinal tract, or on our skin, or on plants. But looking at the various literature that was available at the time, they found that there are 5 major habitats where most microbes live. And these are the different layers of the soil, the kind of close to surface soil, the lower deep continental subsurface soil, but also the ocean and the different layers of the sediment below the ocean. And if you look at this pie chart, you'll see the relative abundance of microbes in these environments, and you find that the ocean, which actually covers a large part of our planet, is a relatively minor habitat compared to the other habitats in the terrestrial in the soil-based structures. Now, these are estimates, and of course estimates can fluctuate depending on the available literature, but it's clear to say that in these environments here, which are in the soil or the sediments below the ocean, there's a huge amount of microbes, and most of these microbes actually live in close association with a surface, so the soil particles, the sand grains, and so on. And whenever this has been assessed, they are also found to produce an extracellular matrix. And this makes them satisfy the definition of what we call a biofilm, which are microbial communities that are surface-associated and produce an extracellular matrix. So biofilms are really the most abundant form of microbial life on Earth, or, like I said, depending on the estimates, sometimes they fluctuate depending on the decade, but they are a highly abundant form of microbial life on Earth. Now, actually, given that microbes, these biofilms are so abundant, you actually are also in permanent contact with microbes. For example, these biofilms are the stuff that's on your teeth when you wake up in the morning. And actually, these biofilms are the reason why you brush your teeth. I always think it's weird that we're in the year 2023 and we still rely on essentially a caveman way to prevent biofilms from building up. We simply scrape them off our teeth multiple times a day. And this is, despite all the advances with our iPhones and whatnot, and we still, scrape them off a few times a day because we have no better way for preventing biofilms from building up. And this is because biofilms are intrinsically tolerant to many stresses. So, for example, they have a high tolerance, around 1,000-fold increased tolerance to antimicrobials, disinfectants, and to other stresses. And this makes infections associated with biofilms very difficult to treat. So here are a list of some of the infections that are associated with biofilms. I don't want you to go through everything here, but just to mention, there's endocarditis, for example, or various tissue-associated infections, but also often implant-associated infections. So whenever you have a catheter, usually you have to exchange it after some time, because it's infected with biofilms, or specifically joint infections, so artificial hips and so on, are infected very regularly. And I know from my wife, who's an orthopedic surgeon, she often implants these artificial hips, for example, and she also often explants these hips because they are often infected with microbes. And once you have a biofilm infection on these artificial joints, then it's almost impossible to get rid of this infection except with surgical removal, because antibiotics really don't work for these implant infections with biofilms. The biofilms are not only bad, they're also good. and we use them, for example, in the treatment of wastewater, to clean the water that comes out of our toilet, for example. And all of these wastewater treatment reactors are based on biofilms in one way or another. This is one particular design here I'm showing you, which is called a moving bed biofilm reactor, where you throw in these small plastic granules here, and they are shown here in higher magnification. And they serve as a substrate for microbial growth. And this yellow thing here is the biofilm. And this biofilm essentially converts the dissolved organic carbon from the wastewater that comes out of your toilet into microbial biomass, which is then macroscopic here. This is on the millimeter scale, you know, or like a centimeter scale. And you can then filter these granules out very easily to leave behind cleaner water. And this is how, at least one of the first parts in how the wastewater is cleaned. So this is one advantage that this is how we use biofilms technologically. We also can use them for production of various amino acids and various chemicals. But another important example of how biofilms benefit our life are the biofilms that form on plant roots. And what's shown here is Arabidopsis. This is a classical lab plant that is studied in the biology community. And on the right here, in very faint red fluorescence, is the root of this Arabidopsis plant. And in green is a biofilm that covers the root of this plant. And this biofilm serves several important purposes for this plant. And this is that it fixes environmental nitrogen, so atmospheric nitrogen, into ammonia that can be used as a nitrogen source for plant, which dramatically promotes plant growth. So it's important for the development for the efficient growth of our crop, our foods that we eat. But this biofilm also trains the immune system of the plant, and in some cases it can also fight away pathogens, invading pathogens that would be detrimental to the plant. Okay, so biofilms are absolutely everywhere on our planet, I've shown you. They're highly abundant. They can be bad in some cases, but also good in other places. They're simply all around us, and we cannot avoid dealing with biofilms. And the question that I have for this talk today that I will discuss is the question of why bacteria actually form biofilms. And to study this question, my lab and actually we realized that we don't have the right technologies to really look at the biofilms from the inside and study cellular phenotypes inside biofilms, which I think is one of the cornerstones of answering this question of why bacteria form biofilms. So to develop these methodologies to look at cells inside biofilms, we developed some microscopy and image analysis techniques that a few years ago that allowed us to look at all cells in biofilms. And this movie stops now. But we can even track cell lineages here. This is shown for different colors, represent different cell lineages. And we can then come up with these family diagrams here, where we can track individual cells. Every cell here is one of these lines here in the lineage tree. And we can then map single-cell properties onto these lineage trees. For example, the cellular position or the cellular level of a particular fluorescent reporter to measure gene expression, or the shape of a cell or the length of a cell and so on. So we have the full information, but what I realized very quickly was that it is these three-dimensional renderings here in these time series are not extremely useful for comparing different conditions or different mutants. Because there you would only be able, in these time series, you would only be able to say like if there's a biofilm or no biofilm, or so, if there are extreme differences. But sometimes extreme differences are important, but sometimes you also want to understand if there are smaller differences that are maybe significant, statistically significant. And so to come up with better ways to compare biofilm growth phenotypes, I would like to propose that this kind of approach here is very useful. So let me go through what this means. So here we take such a biofilm here. This is a three-dimensional object that changes in time, and we have many properties that we can measure for it based on the images. And the way that we compress this dimensionality of this data is to not look at all three spatial dimensions at the same time, but to only look at the principal spatial component. So this is the major variation component in the data. And this, For Vibrio cholera biofilm, that's shown here, this would be the distance of each cell to the center of the biofilm. And instead of using real time on the x-axis, I'm plotting the number of cells in the biofilm, which is kind of a metric of time. But by comparing biofilms of the same cells, of the same cell number with each other, it's actually a better comparison than to compare the time, which different mutants, for example, might take different time to grow. So we have a spatial and temporal graph here, and in color we can then plot various properties. For example, in nematic order, this is the degree of cellular alignment. So my fingers are very highly aligned right now. They would have a high score in this alignment score here, or the single cell growth rate, or the cell-cell distance, and various other architectural properties that we can measure. So we now have a tool to really look at biofilm development in space and in time, and in color we can measure various properties. And this turned out to be quite useful, not only for my lab, but also, I mean, we developed it originally for my lab, but other labs also found this to be very useful. And we couldn't handle all these collaboration requests anymore, so we spun this out into a tool which we call Biofilm Q, which can be then used to analyze these biofilms or colonies of microbes of different sizes. For example, these colonies on agar here, on the millimeter scale, down to more microscopic colonies, or these are Vibrio cholera in yellow in the mouse intestine, which is shown by these blue villi here. All of these kind of structures can be analyzed. And the way that this software can deal with these communities of such a range of different sizes is because it can process two types of data. And the first one is if your microscope doesn't have sufficient resolution to detect all the individual cells, so this would actually be kind of good for detecting single cells, but if your resolution would be a little bit worse, then you might not be able to detect individual cells. And then this tool can segment the community into just saying there's biomass and there's no here there's some biomass and here there's no biomass, and then the tool would dissect this biofilm, this volume, into little cubes, and these cubes can then be analyzed as if they were individual cells, so that you can do cytometry on these cubes. And this doesn't require single cell resolution. But if you do want to have single cell resolution, we also developed a tool for a really highly accurate detection of these single cells. And this is based on a neural network. And the way that these kind of neural networks work, many of you may have already seen this, but let me just go through what this would entail. So this would, to use a neural network for three-dimensional detection of individual cells in three-dimensional images, you would have to take three-dimensional images of a biofilm. So we usually use a confocal microscope for this. This is usually sufficient resolution. And then we would have to annotate individual cells. And this is a painstaking process because you have to annotate in 3D not only layer by layer, as you would in normal confocal images, but you also have to look at the cross-sectional layers, not only XY layers, but also the XZ layers and the YZ layers. And so you have to do really a lot more annotation than you would normally do in two-dimensional images. And this is mentally a bit straining, and this is tough work. I mean, really, it drives people crazy, essentially. Anyway, so we annotated around 18,000 cells in a few months. And then using that data, you can then train a neural network, which I represent by this icon here, like an iPhone icon. And once you have this trained neural network, you can then input these images, put it into this network, and then get out these cell segmentations. And to show you how much better this is to the kind of approaches that we had before, I'm showing you here three different techniques. This is the technique that we previously developed, which was I think the best technique for normal segmentation of individual cells. Here there's a neural network that comes straight out of a box, and here's an improved version of this neural network that we developed based on this STARDIS network. And in magenta color here are false positive detections in this row, and in this here are false negative detections. You can see that there are systematic problems in these normal classical image segmentation, whereas these neural network-based segmentations really are much improved in terms of their reduced false positive and false negative detections. So this is the best tool, and you can then use this tool to input this into BiofilmQ. So BiofilmQ can either deal with this cube-based segmentation or the single-cell import, and then it can do this analysis based on the internal structures of the biofilm. So it can look at all these individual cubes here or cells, depending on what data you have, and analyze things, for example, cellular fluorescence or fluorescence correlations, and so on. And then this tool also outputs various graphs, including these space-time diagrams that I've shown you before. Okay, so now that I've shown you that we have developed these tools, the tool development is actually not the main focus of my lab, but we sometimes realize that we have to develop these tools to answer the question that we have. And the key question, and at least the first part of my talk, is why bacteria form biofilms. So actually, let me stop here. So some examples of why bacteria could form biofilms is this classical example, and that would be that if it is well known that biofilms are highly tolerant to antibiotics, but antibiotics are maybe not the most abundant selection pressure in the environment. I've shown you at the very beginning that biofilms are absolutely everywhere in the terrestrial environment, in the different layers of the soil, where we would expect there to be little antibiotics or other toxins. And so this may not be the most important selection pressure for selecting for this biofilm trait. And so another selection pressure might be predation. Right, that microbes face various predators, for example, bacteriophages. And we, in previous work, my lab has studied the interaction of bacteriophages with biofilms, and that clearly is an important selection pressure because phages are absolutely everywhere in the environment as well. But looking at bacteriophages, I thought that maybe other predators could also be interesting. And one key predator that I then arrived at are the immune cells during infections. So during human infections, of course, bacteria are attacked by immune cells, and so I got my wife to take some samples of human blood of volunteers, healthy volunteers. These are, for example, PhD students in my lab. And they donated some blood very happily, and we isolated various immune cells. So these in red here are neutrophils, here are T cells, natural killer cells, B cells, and macrophages. And in cyan here, this greenish color, are Vibrio cholera cells, and yellow would be the dead macrophages or the dead immune cells. And what I initially anticipated based on our work with bacteriophages, where biofilms were protected from bacteriophages, I thought that maybe the biofilms would also be protected from immune cell predation. But what we found was completely different. It was a completely orthogonal trait. And that was that these biofilms formed around the immune cells in these layers here. After these biofilms have kind of encapsulated the immune cells and become quite thick, then the immune cells would die, and then the biofilms would disperse. And this is kind of illustrated here in this diagram, where this is the amount of biofilm on an immune cell, and this is the time. And these kind of quantifications are then made with BiofilmQ. So this would be to illustrate the usefulness of this. And what we find is that essentially initially they start forming a biofilm, then the immune cell dies around this kind of peak time here, and then afterwards the biofilm disperses and the cholera cells become planktonic again. And for different immune cells, the dynamics vary a little bit, but overall they're kind of qualitatively the same. This is what this phenomenon looks like. So you can see these, somehow these movie, yeah, it's playing on repeat now. You can see that these biofilms form and eventually the cells become planktonic again. Now this is not an unspecific process. It's not that cholera does this on every cell type. For example, if you take the monocytes that we isolate from blood and you differentiate them into macrophages, then you will see that the biofilm formation on macrophages is much higher than on the precursor cells, the monocytes. And similarly also for epithelial cells, this is much lower. Now, we wondered, of course, whether this is kind of an aggregation phenomenon, whether cells just maybe chemotax towards the macrophages, and then maybe aggregate, or whether they actually kind of attach to the macrophages and then grow on the macrophages in situ. And this quantifies this. So we used our single-cell imaging capabilities to define the origin of each cell. on the macrophage, and either define it as being the attachment of a new planktonic bacterium, or whether this new cell on the macrophage surface has has kind of arisen by cell division. And you can see that initially there's a lot of attachment to the macrophages, but then the relative importance, the contribution to the attached bacteria by attachment, it goes down a lot because the cells really grow in situ on the macrophage surface. So they attach initially and then they replicate on the macrophage surface. Now, so this is why we call this a biofilm, because this is a growth-based phenomenon, not just simply an aggregate. Now, we of course also wondered what is the mechanism of the attachment, and luckily for Vibrio cholera, this phenomenon has been, or attachment to various surfaces has been studied, and there are a number of adhesion factors that Vibrio cholera uses, so we knocked them all out here, and we found that flagella are the key for initial adhesion, and MSHA pili are important. And MSHA pili are an interesting finding. These are type 4 pili, because it is known that during cholera infection, humans develop antibodies against MSHA, but it was not previously known by the mechanism by which MSHA could play a role during the infection process. So here could be one mechanism, so this attachment to the immune cells. Interestingly, MSHA pili are not important for epithelial attachment, so they seem to be really specific for immune cell attachment. So then the other hallmark of biofilms is not only that they are surface attached and communities, but also that they possess a matrix, an extracellular matrix by which the cells bind to each other. And so for Vibrio cholera, this is a well-studied model system for biofilm growth. And so due to the work of Fitnat Yildiz and others, we know that there are 4 matrix components that are really important for growth on normal surfaces, so glass surfaces or chitin surfaces or other kind of abiotic surfaces. And these are 3 proteins, BAP1, RBMA, RBMC, and this polysaccharide VPS. And so when you would knock these 4 components out, you would normally expect that actually this strain can no longer form biofilms on glass surfaces, for example, or on chitin surfaces. However, when you knock them out, the parental strain here is able to form as much biofilm on the macrophages as the normal wild-type strain. So these components are not important on macrophages. But then we thought, we just purely speculated that maybe the pili could be part of the matrix. And so when we deleted the MSHA pillars, there was no effect. The toxin co-regulated pillars, this is another type 4 pillars that Vibrio cholera has, also had no effect. But when you start going into double deletions, then there was no there was no more biofilm. And to really test whether these 2 pili are indeed matrix components in these biofilms or macrophages, we used antibodies to localize them, and these are shown in yellow here. You can see that MSHA pili in yellow are really present on the in the biofilm on the macrophages. The toxin co-regulated pillars is also present, and there is another component of the matrix that we found to be very important, and this is a protein called TCPF, which is the most highly secreted protein that Vibrio cholera produces in these conditions. And this is a protein that apparently coats the surface of the macrophages before Vibrio cholera attaches, and then Vibrio cholera attaches with this TCP pillars to the TCPF that it has previously deposited on the macrophage surface. Now that we've kind of looked at how cholera attaches to the macrophages, what matrix they produce to build these biofilms on the macrophage surface, we of course also wondered what the mechanism, or what the function is of these biofilms on the macrophage surface. And this led me to kind of initially speculate that maybe they produce, these biofilms might, because they encapsulate the immune cells, maybe they serve as a diffusion barrier for cytokines so that they inhibit immune signaling. But this hypothesis turned out to be completely wrong, and although I was personally in love with this hypothesis, but it turned out to be wrong. But then, of course, we realized that at the same time as biofilms form, the immune cells die, and we wondered whether biofilm formation could be associated with the killing of the immune cells. And so we looked at how the killing mechanism of the immune cells works and whether biofilms might be important. And so Vibrio cholera is an opportunistic pathogen, and it can produce a number of toxins, actually quite a range here. These are all the toxins that are known that Vibrio cholera has. We deleted all of them, including, for example, this cholera toxin here, CTX. Cholera toxin is the normal toxin that you would associate with a cholera disease, but that turned out to be not important for killing immune cells. And the only toxin that was really important was this hemolysin, HLYA. And when you overexpress it, you induce more cell death here. So how does this hemolysin work? And do biofilms contribute to the action of this hemolysin? So here is the amount of immune cell death for the wild type. And when we knock out the ability to form biofilm, this is the double pilus mutant. So this has a reduced capability to kill the cells, and the hemolysin mutant has also a reduced capability to kill the cells. And when you have both knocked out, the biofilm ability and the hemolysin, you have a further reduction, which tells us that that these two factors have a synergistic effect, that biofilms and this production of this toxin synergistically act to achieve the maximum amount of killing of the immune cells. So how does biofilm contribute to the action of this hemolysin? Well, to find out, we got an antibody against this hemolysin and measured the abundance of this hemolysin around the immune cells in the presence of the biofilm and in the absence of the biofilm. And the antibody is shown in green fluorescence here. And you can see that when the cells produce a biofilm around the macrophages, there's a much higher amount of antibody present compared to when you don't have these biofilms, which tells us that this antibody, the biofilms serve to generate a high concentration of this toxin around the immune cells, and this is the mechanism by which biofilms contribute to killing the immune cells. So encapsulation of macrophages by biofilms increases macrophage death. So to summarize this part of the talk, what I've shown you is that biofilms of Vibrio cholera form on the surface of the macrophages, and how much time do I have left? A few minutes. Okay. Biofilms form on the surface of the human immune cells, of various types of immune cells, both in a cell culture model and in primary cells that we isolated. Biofilm matrix of these particular biofilms is different from other biofilm matrices of cholera, and this biofilm matrix on the immune cells contains pili of this MSHA variant and the toxin co-regulated pilus and this colonization factor TCPF. And these biofilms give a locally high concentration of this hemolysin, which contributes or enhances the macrophage killing. Okay, and now in the last few minutes of the talk, let me move from this question of the functions of biofilms. I've shown you how these biofilms form, but also what the functions of these biofilms are on the immune cell surfaces, to this question of how bacteria form biofilms. And what I would like to show is actually now generalize a little bit away from biofilms and look at microbial communities in general, and maybe look at the simplest microbial community that we have in my lab that we study, and these are bacterial swarms. And the reason why they are so simple is because they are a two-dimensional structure that forms as follows. So here we use Bacillus subtilis as a model system. This is a harmless bacterium, a soil bacterium that in some parts of the world is even used as food. And we would inoculate with a small drop, 25 nanoliters, only a few cells, onto an agar surface. This is a kind of a 10 centimeter size petri dish. And then after some lag time, the cells would grow, and eventually they start swarming out to cover this whole agar surface within a matter of a few hours. And so in the past, we have studied this process by imaging at single cell resolution and tracking the dynamics of this process. And the reason why this is a useful model system for us is because it simplifies the system away from a three-dimensional community, which we study in the context of biofilms, to a two-dimensional community, which is much easier to image, but also to model. And then eventually, after towards the center of the swarm here, a biofilm starts emerging. And so, we now, to really get a comprehensive view of this dynamics of these communities, we don't only image it at single cell resolution. So we have a microscope that is attached to the bottom here that can track the cells at cellular resolution as the swarm expands outwards. So we have the single cell imaging data in space and time. But we also developed a robot to acquire samples in space and time for transcriptome measurement. So we have both the imaging and the microscopy data available. And so this is what this robot looks like. So this is inside a heated incubator on the microscope. These are specialized tips that we constructed for picking up cells. And then this tip drives to this humidity-controlled and heated chamber, picks up some cells from the middle. And this is controlled through a live feedback on this imaging, which is done really at high resolution. And then we know how many cells we picked up. And these cells are then transported to an ice-cooled ice block chamber here with the lysis buffer for RNA preservation. And then we close this tube essentially and perform RNA isolation and RNA sequencing. And because this is robotically controlled, we can really go in at precise spatial and temporal locations to pick up cells. And the outcome are these space-time diagrams, similar to what I've shown you before for biofilms. We have a spatial metric here, the position inside the swarm. This is now on the millimeter scale because these are large swarms, right, that cover the whole Petri dish. And on the x-axis here is time. And we have this at the gene expression level. For every gene, we measure around 4,100 out of the 4,300 genes of bacillus that we can detect here. And we have this phenotypic data. So we have the microscopy information layer at the single cell resolution, right? And so we have these data here, for example, the fraction of non-motile cells, the biomass density, cellular size, and individual cell speed, and so on. And we can now try to build connections between the gene expression with the phenotypic information layer. And so this data is available also if you want to browse through it. It's a massive amount of data, and it actually was quite difficult to find hypotheses that were true, that we could confirm. And because it's such an unconstrained problem, you have so many possibilities for mechanisms that could go on, and many of these mechanisms probably are going on. But of course, the true test is always when you can construct mutants and whether these phenotypes actually play a role. And skipping over a few steps here, what we realized is that when you try to correlate gene expression with phenotypes, one key class of genes are carbon metabolism genes that were highly abundant in the highly correlated genes with the phenotypes. And so here's a map of central carbon metabolism of bacillus. So this is, for example, this is glycolysis, and this is the TCA cycle. This is malate import. This is pyruvate import. And I'm showing you these maps here, these space-time maps, just now very small. And in color here in red are those that are upregulated in the late swarm center, so towards the biofilm state of the swarm. And those labeled with blue font here, the genes are upregulated on the edge, the swarm front. And what we can see is that the TCA cycle is really upregulated towards the center of the swarm, whereas malate import is upregulated towards the front of towards the edge of the swarm front. And malate is the preferred carbon source. And so it seems to be that TCA cycle is upregulated in the swarm center and malate upregulated at the swarm front. And we noticed that also pyruvate intake is upregulated towards the swarm center. And this led us to the hypothesis that potentially the cells perform aerobic fermentation of malate at the swarm front, where the nutrients are highly abundant, and then secrete pyruvate. And then at the swarm center, they take up pyruvate again to fuel the TCA cycle to generate energy. So we think that energy metabolism might be different in these two regions, at the swarm front and at the swarm center. So can we test this? Well, we did the same assay now to test this hypothesis, but not sampling cells for transcriptomes, but acquiring cells, acquiring the agar and measuring metabolites. So we now regularly sampled here the agar and analyzed the metabolite content within the agar. And we found that initially the malate is actually consumed, similar to what we had speculated, of course. And so malate is consumed first on the region towards the center here, and then the red stars here would be this line. Then malate is consumed here, and then towards the outer region, malate is also consumed. Other sources might even be more preferred here, succinate actually, but other metabolites displayed a different pattern completely, and this was shown for pyruvate and a few other metabolites that were initially secreted by the cells that were using malate and succinate, and then this pyruvate is then consumed by cells that migrate over this location at a later point in space and time. So pyruvate is really a cross-fed metabolite between different space-time regions. Right? So the cells that secrete pyruvate are not the cells that consume pyruvate. So we've really shown this kind of spatio-temporal cross-feeding of metabolites. And with that, I'd like to thank the various people who did the work here. So Hanna, Jekke, and Niklas and Takuya did this work on image analysis a lot. And Lucia Vidakovic, who just graduated, she left the lab after graduation to join the company BioNTech. She did the work on the macrophages that I presented. And Hanna was also the person who did the work on spatiotemporal cross-feeding and swarms and the spatiotemporal transcriptomes. And with that, I'd like to thank you for your attention.